Hey guys, welcome back. Another e-learning day, I guess very similar to yesterday. Today we're looking at 10.3, which is independent assortment. I'll try to keep these notes as short and sweet as possible for you, since I know we've had back-to-back e-learning days here, and that probably isn't the funnest thing in the world. But hey, at least we're not having to wear a mask. So 10.3. Independent assortment. So this is a law also discovered by Mendel. So our Austrian monk, Gregor Mendel, using just his pea plants. Again, he didn't know about DNA, he didn't know about chromosomes, he didn't know about a lot of the stuff you already know. He was able to determine So again, Mendel was working with his pea plants, and simply by observing their phenotypes, when he crossed them, when he mated these plants, he was able to determine all three laws that we use today to govern genetics. So his third law is what we call the law of independent assortment. And keep in mind, he figured all this out without knowing about meiosis, which you guys learned about yesterday, without knowing about chromosomes, without learning about DNA. So it should hopefully make pretty good sense to us since we know how meiosis works. But again, he didn't. He was able to figure all this out without that. So what the law states So what the law states is that the alleles of two or more different genes get sorted into gametes independently of one another. That's the independent assortment. They get put into gametes independently of one another. So to kind of illustrate this, let's take a look at an individual. So we'll take a look at an individual heterozygous for two genes. We'll call those genes gene A and gene B. So heterozygous means they have one dominant and one recessive allele. So here's their cell. We'll say that they've already been through S phase. They've already copied their DNA, so it's already in that X shape. And here's the key. So far, we've only looked at one gene, or assuming just kind of one chromosome. But you have multiple chromosomes. We'll say this individual has two chromosomes. Again, if you keep in mind, humans have 23 pairs of chromosomes. This isn't so hard to believe. Now again, they're heterozygous for these chromosomes. So that means they have big A. Now this one over here is also going to be big A. That is an exact copy. Remember, that's the idea. We have two sides of the X. They're both going to be big A because those are exact copies. But the homologous pair over here, the homologous chromosome, is going to be little a. So for gene A, this individual is heterozygous, one big, one little. And then on the green chromosome, we'll say that they have B. And again, the two sister chromatids are going to be identical. The two sides of the X, those are going to be identical. So if you have big B here, you're going to have big B on that one. But heterozygous means that the homologous pair of the other chromosome is going to have little b. All right, so we are pretending that here during meiosis 1, they line up like this. And so when they separate, which is what's going to happen in meiosis 1, we're going to end up with the dominant alleles together and the recessive alleles together. We go through meiosis 2. And we produce the gametes big A, big B. Oops, excuse me, sorry about that. So we've got the gametes big A, big B. These are both going to be like that. And the gametes little a, little b. So we have gametes A, big B, and little a, little b. But here's the idea. There is no rule way back up here in meiosis 1. There is no rule that says that they have to line up so that the big letters are on one side and the little letters are on the other. There is no rule that the dominant letters stick together, the dominant genes stick together. In fact, not only is there no rule, but Mendel's law of independent assortment states that the rule is that they don't have to line up like that. Can they line up like that? Sure, nothing against that, but it's also very, very possible that here in meiosis 1, and this would generally be taking place during metaphase of meiosis 1, when the tetrads line up on the metaphase plate, we could also get this combination. So I'll make my A's line up in the same place. So I've got the dominant letter on the left, the recessive letter on the right, but the B's I'm going to flip. I'm going to put the recessive letter on the left and the dominant letter on the right. And again, Mendel's rules are that can happen. In fact, that should happen 50-50. It's completely independent. The way that the A allele lines up is completely independent. It has no influence on the way that the B alleles line up. It's not like they say, oh, all the dominant alleles over on this side and all the recessive. Nope. It's perfectly possible that you pass on one dominant and one recessive, as we'll see here. Again, we'll go through meiosis 1. Well, that's going to be problematic when I bring the arrows down. Go through meiosis there. So we can already see we've now separated our chromosomes to have one dominant and one recessive. One dominant and one recessive. So when we go through meiosis 2 then, and we form these gametes, it is possible that you have gametes with some dominant letters and some little. So we've got gametes over here, all dominant and all recessive, and we have gametes over here that are dominant and recessive. So we've got all these different combinations of alleles. Again, there's no rule that says the dominants have to go together and the recessives have to go together. They can, but they sort independently. It's a 50-50 chance, depending on how they line up here during meiosis 1. And that's the rule of independent assortment. Mendel's law of independent assortment is that dominant letters don't influence the other dominant letters. All of these are equally likely, 25% chance that any one of these 4 gametes would be produced. Hey, we're going to work more with this next week, but that's all for today. You have a homework that goes along with this, so make sure you guys check Canvas for that. And I'll see you guys on Tuesday. Have a good weekend.